Okay, so if you recall last week, we went over, and I kind of went over what the test was going to be on. So I'm just going to do a little review right now of 18.4, which is prokaryotes. Okay, so remember that prokaryotic cells are controlled by operons, which are groups of genes that are controlled by one promoter. And so remember that there were two types of operons that we talked about. We talked about the trip operon. Trip stands for tryptophan, remember? And what it does is this controls genes that make tryptophan. So this makes tryptophan, or makes the enzymes, I should say, that make tryptophan. So remember, let's go over this. So here's trip operon. So here's your DNA, and remember your book has these little things here, which means that you have there's a gap, and so the DNA is much longer. So my line is a DNA, and so remember that there is a regulatory gene upstream. All right, so this is the regulatory gene. And then there is the promoter region. So the promoter region, remember, has two parts. And so it has the operator part, and that's part of this. So if this is the beginning of the gene, and this is the end here. So what happens is you have this operator, and your book kind of showed you it in terms of a particular shape, and so the operator region has a particular shape that kind of looks like this. So if we kind of make the DNA kind of a block like this, all right, so kind of disregard that center line. So this has a specific binding site, and the operator region is part of that promoter region. And then after the promoter region, you have your genes. And there are 5 genes, A, B, C, D, E, D, C, B, A. And these are called trip A, trip B, and so on. All right, so that's what we have here. And so what happens is this regulatory gene called trip R, for regulatory, can be transcribed, makes an mRNA, and then the mRNA is used to make a protein. In this case here, the protein is a repressor, but the repressor is inactive. So what happens is we have this. I'm not the best artist here, so I apologize here. So that's what your repressor, and it's in the inactive state, meaning that it can't bind to the operator. So that's what that means, can't bind to operator. So the repressor represses or turns these genes on, all right? And this whole thing, from the promoter region to the end here, that's called your trip operon. So if this can't bind, then that means that these genes are normally on. So therefore, RNA polymerase, I'll do it in a different color here, RNA polymerase is able to come and bind to the promoter region. RNA poly, I'll just put. And it is able to transcribe the gene. So as it moves here, what does it do when it transcribes it? So transcription, it's making a long piece of mRNA that is coding for all 5 of these genes. So this is your mRNA. And this now can go to your ribosome to do translation and makes enzymes to make tryptophan. I'll just put trip for tryptophan. And so therefore, because the repressor is made in the inactive state, this operon, is it always on or always off? All right, as it's made, as it stands right now. It's always on. So it's usually on here. So this is usually on, which means then it is able to be repressed or is able to be stopped. And when would you want to stop this? When would you want to stop making tryptophan? When tryptophan is present. So this, let me pick a different color here. So this will be turned off when tryptophan is present. And the thing is that it's usually on and it's able to be turned off means that this is an example of a repressible operon. So this is a repressible operon because of that. So what happens when tryptophan is present? So when tryptophan is present, if we go back to this picture here, when tryptophan is present, what it does is, if this is your inactive repressor, okay, so this is inactive, sorry, repressor. It becomes active when tryptophan is present. If there's tryptophan present, it becomes an active repressor. So what does that mean? Tryptophan can bind to it. So here's tryptophan, binds to it, and changes the shape. And so now the shape and tryptophan is bound to it. So there's trip. Now it's active. So what does that mean? Now this can bind to the operator. So we go back to up here. So if it binds to the operator, so here I'm going to go ahead and I'm just going to, if it binds to the operator, well, maybe I'll just no, I don't need to redraw it here. When it binds to the operator, it comes up here. I'm going to change shape here. And if it binds to the operator, so here's your active repressor. Genes are turned off. Are off. Why? Because RNA polymerase physically can't get to the genes to transcribe them to make the enzymes to make tryptophan. And that's when we want the genes to be off, because it would be a waste of energy and resources to make tryptophan when the bacteria is getting it regularly through its environment. So when there's tryptophan present, the repressor is activated. And so therefore genes usually turned on and turned off when the repressor becomes activated. All right, so the repressor is made in the inactive state, which makes it usually on. So that's your trip operon. All right, so it's a repressible operon. So then we have one other operon that's different, and that was the Lac Operon. So the Lac Operon is different in that instead of making something, all right, so here was making tryptophan, the Lac Operon digests the sugar lactose, which remember is a disaccharide. So here's your Lac Operon. So here's your Lac Operon. Remember, you have a regulatory gene. So here's your DNA. Now we could have some space in between here. So this is, I did it again, just drawing a straight line. I should do it like in a box like this. So this is your DNA. This is your regulatory gene. And then you have your promoter region, in which, and I should show here, in which the operator is part of that promoter region. And instead of the trip genes now, we have 3 Lac lactose genes, lactose Z, Y, and A. So this is what we have going on in the operator, or the operon. So this whole thing right here, from the beginning of the promoter region, from here to the end here is called the Lac Operon. It's controlled by this regulatory gene. So it makes mRNA, but that makes a repressor. But in this case here, the repressor is made in the active form. What does that mean? If it's made in the active form, guess what? It can bind to the operator. And so that's what we have here. And therefore, because of that, RNA polymerase comes over. So here's RNA polymerase tries to bind, and RNA polymerase can't bind. What does that mean? Genes are usually off. And when would we want these genes to be turned on? Well, what do these genes do? These genes, these three here, code for enzymes to digest lactose or to metabolize lactose. And so we would want these genes on only when lactose is present in the environment. So if lactose isn't, then the genes are off and we don't make these enzymes. So then if the bacteria's environment changes and there is lactose, all right, so if lactose is present in the environment, then what happens? So if lactose is present, then what happens is we go from active repressor to inactive repressor. And so what does that lactose do? It binds to the repressor. So here's the active repressor like this. It's active. And actually it's, remember the isomer, allolactose, that's only present if lactose is present. So that tells that lactose is present. And so it binds to it, and when it binds to it, you get, from the inactive repressor, you get the shape change. And so now the repressor, it's the allolactose, now it's in the active shape, and this can't bind to the operator. So looking at here, this shape right here will not allow it to fit into the operator up here, and then therefore the genes, oops, my lights just went off. Okay, the genes are turned on or off? On. You should have the genes are turned on. And therefore now, what are you going to do when the genes are turned on? RNA polymerase, this is not going to be bound here. RNA polymerase is going to transcribe this and therefore make the enzymes to digest lactose, which is what you want when lactose is present. Okay? And so that's the Lac Operon. Now both of these, the Lac Operon and the Trip Operon, are considered, I'm going to put here, what's called negative control. Because it's the repressor that will repress the genes. What we're going to look at next is moving on with the Lac Operon, when the Lac Operon also gives an example of positive control. So glucose versus lactose. If both of them are present in the cell, then bacteria prefer glucose. And so therefore, you want, when glucose is present, you want the Lac Operon off. You don't want to make enzymes to digest lactose when you can use glucose. All right, so how does it do that? How does it, if glucose is present? So what happens is we have this cap protein. And so the cap protein is going to help turn this Lac Operon on or off depending upon if glucose is present. If glucose is absent, and I should say glucose present and lactose present. So they're both present, so you have a choice, and we want to use glucose and not lactose, so we want the Lac Operon off and not make the enzymes to digest lactose. Well, if glucose is absent and lactose is present, we want the Lac Operon on so that we do make the enzymes to digest lactose. So we're only making and turning this on, on. I don't know why I thought I was thinking on here. On. That's on. Lac Operon on. So that's what we want. So we use this cap protein. Cap is normally in the inactive form. So this is your inactive cap. So what happens is it becomes activated when something called cyclic AMP binds. So this is cyclic AMP. Cyclic AMP, remember, is only present when glucose is scarce. Remember, it breaks down ATP into cyclic AMP. Cellular respiration isn't happening. And so that's only present when glucose is scarce. So then what happens is it activates the cap. And so you still have your cyclic AMP bound here, but now your cap changes shape. So this is kind of like what your book shows here. So this is your active cap. And so what happens is this then binds to the promoter. And then RNA polymerase binds and transcribes the genes. Transcribes genes, and operon is on. So that only happens when glucose is scarce. Well, remember, so glucose absent, so that's what we're looking at here. So this right here, glucose is absent. And so then this cap, if you look at your figure in your book, that's what this picture, I have this picture here. So it's binding here, binds to the promoter. RNA polymerase can bind, the genes are turned off, but remember that lactose is present too, so you have your inactive repressor, and so there's no repressor bound, and so therefore the genes are turned on. If, for instance, the glucose is present and lactose is present, if glucose is present, then you're not going to have any cyclic AMP, and this cap isn't going to be activated, so it's going to stay in the inactive form. So you have inactive cap, but lactose is present, so then your repressor is inactive as well, but RNA polymerase cannot bind, all right? And so that's when the glucose is present, and so therefore you're not going to make the enzymes to digest lactose because you have glucose to digest and use. And so therefore it's not turned on. So even though, with Lac Operon you have negative control, the use of a repressor, but you also have positive control, meaning that in order to turn the Lac Operon genes on, you have to have an active cap protein. And so those two things have to be true in order to turn the gene on. Active cap, inactive repressor. And so that shows both positive and negative control. And that's basically the control in prokaryotes. I'm going to do this video I'm going to do it in sections. So I'm going to do eukaryotic gene expression in a separate video. All right, so look forward to that.